Antibiotic resistance is one of the most dangerous health problems that modern medicine is facing. Scientists have predicted that by 2050, 10 million lives per year could be at serious risk of developing antibiotic-resistant infections. Since 2008, the NHS has been concerned about E. coli bacteria becoming resistant to antibiotics due to over-prescription of antibiotics when treating coughs and colds, which are minor infections that could be healed naturally. According to recent studies from the universities of Oxford and Bristol in 2016, 80% of all urinary tract infections in children were caused by E. coli. When infectious bacteria were tested, it was found that they were multi-drug resistant. As an antimicrobial agent, Hepcidin could be used to tackle this global issue. Hepcidin causes a reduction of extracellular levels of iron, which is a form of nutritional immunity. Further studies can be carried out to test the antimicrobial activity of recombinant hepcidin against extracellular iron-requiring pathogen, Vibrio cholerae, which causes cholera. Properties of hepcidin. It is a 25 amino acid peptide hormone that weighs 2.8 kilodalton. It maintains iron homeostasis and is encoded by the HAMP gene, which is mainly expressed in the human liver by hepatocytes. Mechanisms of hepcidin. Hepcidin 25 binds to ferroportin, which is a membrane protein involved in the movement of iron in mammalian cells. This induces the internalization and lysosomal degradation of the hepcidin ferroportin complex, which inhibits the release of iron into the plasma from liver macrophages. Hepcidin fights off extracellular iron-requiring bacteria, such as E. coli, by reducing extracellular levels of iron, which is known as nutritional immunity. Hepcidin can be produced on a large scale by making E. coli express it. To enable purification, the gene is fused with a fusion partner. This is a short amino acid sequence which has special properties. When designing the vector plasmid for transformation, the gene encoding hepcidin, called HAMP, was linked to an elastin-like recombinant tag using restriction enzymes. These were cloned into a plasmid which also contained an antibiotic-resistant gene, for example, ampicillin. E. coli bacteria are made competent using calcium chloride and are made to take up the plasmid through a heat shock procedure which sweeps the plasmids into the cell through the pores called adhesion zones. These cells can then be selected for using ampicillin. The cells are cultured until a significant number of bacteria is obtained. They are then induced to express hepcidin in large quantities. The cells are lased with sonification, which uses ultrasound to break plasma membranes. The elastin-like recombinant tag enables an easy and cheap method of purification. At low temperatures, the molecule is soluble, so when the lysate is centrifuged, it remains in the supernatant. Next, this supernatant is heated, which causes the tag to self-assemble. This time it will be in the pallet after centrifugation. As this cycle of hot and cold centrifugation is repeated, all contaminants are removed. This is a simple procedure called inverse transition cycling and yields pure hepcidin. The tag can then be removed using formic acid, releasing the antimicrobial peptide. Its properties can be tested using disc diffusion. When hepcidin is placed in a plate covered in E. coli, it generated an inhibition halo, which demonstrates its antibacterial properties. As bacteria become increasingly resistant to classical antibiotics, the demand for alternative antibiotics such as hepcidin increase. As this peptide is naturally produced in the human body, it is predicted to be a safe drug for human use. Producing hepcidin fused to a tag with special thermal properties allows it to be produced and purified cheaply. Research indicates that hepcidin is not only antimicrobial as it has potential anti-malarial properties. The production of hepcidin through recombinant DNA technology is potentially beneficial to global health and deserves the attention of the scientific community.